In this video, we're going to be looking at transcription. So we're going to look at this first part of central dogma, right? We're going to be looking at transcription, which is the transcribing of the DNA code into an mRNA molecule, all right? And then to complete the central dogma, right, the mRNA molecule is translated into proteins, is what we're seeing here. All right, so we're going to focus in chapter 3, we're really looking at this. All right, that's really what we're interested in. All right, so a quick review of DNA. All right, we want to remember we have a 5' end and a 3' end based upon the presence of a hydroxyl group at our 3' end here and a 5' phosphate at our 5' end. So we have a directionality, all right? We also want to remember that there are two strands and they are anti-parallel to each other. So we've got a 5' to 3' running strand over here and a 3 to 5 running over on the right. Now towards the center of our double helix, right, so we should remember that this thing looks kind of like this, right? It kind of looks like a spiral staircase, all right? Towards the center then we have oriented these nucleotides, or the bases for our nucleotides, and they are present right here and here. We have base pairing rules in DNA, right? It's always A to T and G to C. All right, so we will see equal amounts of A as T, all right, and G and C. So let's say that 40% of our DNA was A to T, well then 60% would be G to C, right, for a total of 100, all right? And these, the G and the C, would be each 1/2 of that 60%. All right, so these things were running anti-parallel, and we have our bases towards the center, and they are hydrogen bonding. All right, so we've got a G to a C here, A to a T, A to a T, G to a C. Okay, so we have more hydrogen bonds between the G and the C. Now the other thing that occurs is that we have some hydrophobic interactions between the bases going in this direction because of the ring structures. They tend not to be extremely polar, and the polar parts tend to be involved in the hydrogen bonds. And so that allows then this stacking, base stacking attraction of sort of compressing and holding these bases together from top to bottom. All right? Okay, so the parental strand of DNA, which is our dark blue here, all right, it serves as a template for daughter strands. So by template, we mean that if this is my template strand, so let's look at it going this way from left to right, all right, that every time there's an A, you put a T in the daughter strand. If there was a G here, you would put a C here, T here, A here, and so on, right? So it provides a template for the building of the daughter strand, all right? So some rules, all right, and this is true for DNA replication and transcription, all right? So these rules are that you read DNA, so the polymerase moves along the DNA, 3' to 5', all right? We synthesize, so synthesis of DNA or RNA is 5' to 3', meaning that you're adding them onto this 3' hydroxyl end. All right? Now, the DNA is too long to actually fit inside of our cell, so it's supercoiled. And so what we've got here is demonstration of somebody twisting on a piece of string. All right? And so it's being shown that they're twisting this in this direction. Okay? And what's happening is that it's introducing supercoil, so you're getting these knots basically, all right? And so to package the DNA and to get it to fit, much like all the string inside of a baseball, or the rubber bands inside of a golf ball, if you've ever torn those apart, it all has to be wound up and sort of packaged in a mechanism in a means that allows us to fit this massive molecule into a very small space. So there are histone proteins that are used to assist us in this. Okay, so they are histones. And what happens is the DNA is wrapped around, so we've got this double-stranded DNA, and it's going to be wrapped around these histone proteins, and that's going to help us twist and supercoil and wrap all of this up into chromatin, which is then packaged into the nucleus. All right. So we're not going to do a lot of detail on this. You can learn more about this in the molecular genetics portion of genetics, but I do want you to realize that there are proteins that are used to help us wind up the DNA and package it so that it fits. All right, a couple of other things that we want to talk about, all right, before we get going on transcription, is that here we have uracil and over here we have thymine. And you'll see that there's a methyl group, all right, on the thymine and a hydrogen on the uracil. So they're pretty similar all told, all right? The reason it's believed that uracil is used in RNA is that it is a breakdown product of cytosine, all right? So if you took cytosine, all right, it becomes uracil. Well, because uracil is not supposed to be in DNA, it's easy to repair this, right? When it degrades, when the cytosine degrades to uracil, it's easy for an enzyme to recognize this and repair it. If there was uracils already present in the DNA, then you would introduce a mutation. You wouldn't know which uracils to replace, right? Which ones were the degradation products of the cytosine. All right, so that's why we believe that uracil is only used in RNA and not in both. All right. All right. So what we're going to do in terms of transcription, right? So this is a picture of transcription. Okay. So let's have a look. We've got 5', 3' on our two DNA strands, right? So this one is 5 to 3 this way. Okay. Wait a minute. It's interesting. They've got a typo here. It's showing it as 5 to 5 and 3 to 3. So in reality, this one should be 3'. This one should be 5'. All right, and this one, yeah, stays as 5'. Okay. So remember, we're going to read, right, our template 3 to 5. So that's this one. Right? We're going to read it in this direction. All right. We're going to synthesize. All right. Let me change colors here. Going this direction, and you'll notice it's 5 to 3. So this is synthesis. Now, we have a template strand, which is the strand that is used to make our RNA transcript. And then we have the complementary strand of DNA that we call the non-template strand. It is not used in this case. All right? All right. So let's look at some DNA. So we're going to simplify our diagram a little in terms of the way the DNA appears. All right? And what we want to look at is a couple of things. All right? First, let's look at the promoter. All right, so the promoter is the RNA polymerase binding site. All right, a series of proteins will bind to this promoter. All right, and it's just a sequence of DNA. Right, so the promoter is a DNA sequence. All right, that is going to allow for the binding of general transcription factors. All right, that will then allow for polymerase 3 to bind. So this is the site where polymerase 2 in eukaryotes to bind. Okay, so all of this stuff happens at the promoter, all right? Now, there will also be what we call activator proteins, okay, or an activator protein, and we'll get to that in a little bit, all right? So an activator is also going to be required, okay? So if we look at our diagram here, we've got a promoter here and a promoter here, all right? This promoter goes in this direction, all right? So we're going to, our transcript, 5' to 3' means that we use the top, right? Because we go 3, notice we're 3 on this end, so we're going 3 to 5 in this direction. So we use the top as our template strand, all right? If we look over here going this direction, all right, remember we synthesize 5 to 3, we read 3 to 5. So we start here, read in this direction, okay? Now, so the promoter lets us set where we're going to start transcribing our gene, right? So that we can transcribe it into RNA, then that RNA can be translated. Now, when we get to sort of the end of our gene, all right, we have a terminator sequence, which is a specialized sequence of DNA that causes the transcription to stop, and the mRNA is then released and can go on its way to being translated. Okay. So what you should see then is that both strands of DNA serve as templates for transcription, but they function in opposite directions, meaning that the bottom one, 3 to 5 being read this way, synthesized 5 to 3 this way, the top, 3 to 5 going in this direction, synthesis 5 to 3 in this direction. Okay, so we use both, all right, but they go in different directions. All right, so here's my promoter site, all right, and then here are my enhancer sequences. Okay, so let's have a look at a couple of things, all right. Within the promoter, all right, so I'm going to draw this in green. Okay, within the promoter is a sequence called the TA-TA box. All right, and what it is it's a sequence, and if this is my 5' end and this is my 3' end, all right, it looks very much like this often. So it would be TA, TA, or similar to that. Okay, now the promoter, right, is, you know, a few hundred base pairs in size. All right, the proteins and the polymerase are basically going to lock into place at that ta-ta box. So we're going to have this protein and these other associated things, and they're all going to kind of lock into place. And I've drawn them too large, right, because my promoter isn't that big. But the basic idea is that there's a series of events that is going to occur at our promoter. All right. Now, at the enhancer sequences, remember I mentioned that we needed activator proteins. So enhancer sequences are away from the promoter some distance, all right? And what happens is that these enhancer sequences of DNA recognize activator proteins, which are shown here. These activator proteins will cause the DNA to fold up in a manner that allows the general transcription factors and the RNA polymerase to bind, okay? And then we're off to the races, so to speak. All right, so here, all right, here's the enhancer sequences. Here's our activator proteins. Here's my promoter, all right? Here's the mediator complex, which is really the entire thing. It is the transcriptional activator proteins, the RNA polymerase, and the general transcription factors, right? They're all brought together, and that forms this mediator complex, this whole thing. We'll just consider it that way. Now, At this point, then, we can actually have transcription occur. All right, so here's our RNA polymerase, right? This big thing, and it's moving along the DNA. So this top, if it's moving in this, it's moving in this direction. So the top one is our template strand. The bottom is the non-template strand. All right. So what we do is we create a bubble. So we're going to split or unzip the DNA, right? So we've unzipped it, and we are adding the bases to the 3' hydroxyl end, right? So this RNA transcript is growing in this direction, right? So we're adding them on here as we unzip and move. We always add them to the 3' end in the 5 to 3 orientation. So let's look a little bit more closely. Here's my template strand, 3', 5'. My RNA transcript, 5 to 3. So we're building this thing, we're adding on here, we're reading this in this direction, okay? Now, we're using complementary base pairing, all right? So that's what's being shown here, okay? And what we're doing is we're adding, all right, a nucleoside triphosphate, all right? So there are 3 phosphate groups on my new nucleoside coming in. We're going to cleave 2 of those phosphates off. This basically is providing the energy for the reaction to occur. All right, and we're going to add then this 5' phosphate at the 3' hydroxyl to make our phosphodiester bond. All right, so we start with the high energy nucleoside triphosphate. And you can call it a nucleotide if you like. Triphosphate. Okay, so that nucleoside triphosphate is going to be bound, and it's in the 5' to, right? This is its 5' end here. So it's in the 5 to 3 orientation, and we're basically going to take this thing and attach it right here. Okay, and the only nucleoside that can fit in here is one that is complementary to this base here, right? So if this is an A, this is a U, all right? Now, if we look, right, so this is my RNA polymerase here in green, all right? Here's my DNA, right through here. Here's this bubble that we saw that was created. So what we're doing is we're breaking the non-template strand away, right? And we're not really using it at all. The template strand goes into our polymerase, into the active site where we're going to actually catalyze the addition of the trinucleotides. All right? So here we have all of these trinucleotides that are funneling into this reaction area, all right? And we're adding these bases 5 to 3. Now, temporarily, we have an RNA-DNA duplex. So we can think of it as, so we've got DNA, we've got RNA. Well, what's going to have to happen is we're going to have to split that apart. And that splitting occurs right here. All right, so where we break the hydrogen bonds, we peel that off, and then our RNA transcript goes out this way. That allows the 2 single-stranded DNAs from our bubble to come back together, and our DNA is left intact. All right, so let's summarize what we've done. All right, what we have done is, if we think about, we've got this 1 strand of DNA, 2 strands of DNA. All right. Now on this we have a promoter region. Okay, so I'm going to put this here, and I'm just going to put P. All right. We also have an enhancer region. All right. I'll put this somewhere, and it can actually be downstream or upstream. It doesn't really matter. Let's just put it here just to make it simple. All right. Now in heat downstream from all of this is my gene. All right. So I'm going to make this 3', this 5', 3, 5, and that way my promoter is going to go in this direction. Okay? Now, so what has to happen at the promoter, right? What needs to happen here? So we need these general transcription factors. Need to bind. Then the enhancer proteins come in, all right? So the enhancer proteins, right? And these bind to, so we got to remember the promoter is a DNA sequence, right? The enhancer is an enhancer sequence, all right? And then the general transcription factors, the enhancer proteins, they bind to those DNA sequences, okay? And then the enhancer protein, general transcription factors, then what happens is we get the polymerase. And its associated proteins. Okay, and then we get the mediator complex, which is sort of occurs after we get this interaction between these two things here, the enhancer proteins and the polymerase. All right, so general transcription factors, then we sort of in sync with each other. The enhancer proteins, the polymerase, associated proteins bind to the promoter, and then the mediation complex. Remember, all of this being basically the recognition of the TA box, and approximately 25 bases downstream from the TA box is where the very first nucleotide of our RNA molecule will be added. Okay? So some things to practice doing are taking a piece of paper or a whiteboard and drawing out some double-stranded DNA. All right? And then you can label it 5, 3, 3, 5, right? You want to make an anti-parallel. And now what you want to do is really kind of what we did on the last page, right? You want to come up with a promoter region, all right? And so it's here, all right? If we're going to go in this direction, right? Maybe it's over here if you want to go in the opposite, just so you can fit everything in. All right, and so remember, you're going to read the DNA 3 to 5, which puts us on top here, bottom here. All right. You want to think about the enhancer. You want to think about the sequence of events. All right. You may even draw some things on here. So you might put some, proteins here that would be our general transcription factors. Then you would bring in your polymerase, right? This big thing. And then you would, we need our enhancer, right? And so we've got an enhancer protein, and it's linking over to this. And then we have some mediator protein that, the mediator complex. Sorry, not just an individual protein, but our mediator complex. So it's a number of proteins, right? All of this stuff binding in place. All right, and then you can start to think about some of those details of the actual addition, right, of all of the bases. And then down here somewhere we have to have a termination sequence. All right, so you want to walk through all of that. So we've kind of got initiation, which would be the adding of all the transcription factors, the polymerase, all of that. Then you would have elongation, right, which is where it just kind of motors along, synthesizing the RNA. And then you have termination, which is where it ends. Okay, and we're not really getting into too much detail on how termination occurs, other than it terminates at a specific sequence. Now, in the bacterial world, things happen a little bit differently. So this is our prokaryotic cell. We have a circular chromosome, all right? what we're going to do is we are going to synthesize our RNA 5 to 3, okay? Use a promoter. Enhancer sequences are not always required, all right? Sometimes there's an activator, what's called an activator, sometimes there isn't, all right? But what we use is, instead of the general transcription factors, the bacteria use something called sigma factor. All right, and there's a number of these. All right, and the sigma factors are specific for the genes that they transcribe. So there's a sigma factor for housekeeping genes, right? And housekeeping genes are always turned on, they're always being transcribed. They use that sigma factor. Other groups of genes use different sigma factors, all right? But just as we did before, you'll want to, right? The main difference is that you've got your 2 strands of DNA, all right? You've got a promoter, all right? And in this case, it's the sigma factor that helps recognize the specific spot within the promoter where the RNA polymerase can bind and we can get started, okay? The other thing that is unique in the prokaryotic cells is that because there's no separation between the nucleus and the cytoplasm, right, that we see in the eukaryotic cell, the mRNA actually heads right on out into it's synthesized in the cytoplasm where there are ribosomes, and the ribosomes then can instantly start translating it into protein, okay? And since that is the case, that allows the bacteria to respond very quickly in terms of transcription. The other thing that this should suggest to you about the bacterial mRNA is that there's no editing required, right? It goes straight to the ribosome and is translated into protein. Whereas in the eukaryotes, we're going to have to do some editing, and that's what we're going to talk about. All right, so this slide is showing us editing. So what we've got is we've got our DNA. Oops. All right, so we've got our DNA here, okay? And there are coding and intervening strands here. And so we call the coding strands are called exons, and the intervening and non-coding strands are called introns. So we've got DNA here. It's transcribed into what is called the primary transcript. All right. The primary transcript gets a 5' cap. The 5' cap is a methyl guanosine. All right, so I'll just write this here. Methyl guanosine. So you want to think of it as it's a specialized G. Okay. So that's put here on the 5' end at the cap. All right. At the 3' end, a poly A tail is added. So a series of A's, a long series of A's, is added of about 250 A's, right, compose that poly A tail. Now, what's going to have to happen then is that, so we've got a cap, we've got a tail. That helps stabilize this RNA molecule. Then specialized enzymes are going to come in, and they are going to splice or cut out the introns. All right, so the introns are cut out, and then we're going to splice the exons together. So we're going to cut the introns and then link together the exons, the coding regions, and now we have our functional mRNA that's ready to go. Okay, the complex, or the enzyme complex that does this splicing is called the spliceosome. And we will not get into the specifics of how the cutting occurs in the spliceosome, but what I want you to know is that the spliceosome does the excision and splicing, all right, and it's made up of both protein and RNA. All right, so the spliceosome is a complicated structure. All right. Another thing so this is just a picture of our cap. Sorry, I was jumping ahead. All right, so here we can see the 7-methylguanosine here, and it's linked in a 5 to 5 phosphate linkage, and so we're trying to protect this, and it's a special linkage here, and it's basically a protective cap at the 5 prime. Now, the other thing that can occur is alternative splicing. All right. So if we look here, we've got introns in the light blue, exons in the dark blue. Okay. So we have 3 exons. All right, and we've got a little 4th one down here. So in alternative splicing, what happens is that here's our primary transcript, right, with all 4 exons. Well, protein A is a combination of E1, E2, and E4. So these 3 exons make protein A, whereas 1, 2, and 3 make protein B. And so one gene then led us to 2 different proteins based upon alternative splicing. And this is very common in eukaryotes. Okay, so if you were to look at this, this lecture, what you want to take home in terms of big concepts is that you want to be looking at DNA and RNA structure in terms of how they allow the process of transcription to occur. Okay? Then we want to look at the transcription, and you want to be thinking about the orientation, right? We read 3' to 5, you synthesize 5 to 3. You want to be thinking about promoters, enhancers. There are proteins that are associated with them, right? Key proteins. And all of this stuff, right? This would be kind of initiation. And then we would have elongation, termination, and then splicing. Right? So there's a number of things there that you want to make sure that you've got and that you can do those in a logical progression.